Hello everyone, welcome to my channel. Today in this video, I am going to discuss about meiosis. In the previous video, I have discussed about mitosis, cell cycle and all I have discussed. So in this video, I am going to start meiosis. Before that, I am going to explain what is the difference between mitosis and meiosis. So, mitosis will take place in somatic cells. Now, what is somatic cells? Somatic cells, all the cells present in our body except the gamete cells, that is sperm and egg or ova. Other than that, all the cells present in the body, skin, liver, kidney, all the organs made up of cells, all the cells will be somatic cells except the gamete cells. Somatic. So these, there in the somatic cells, mitosis will take place, and the number of cell, the parent cell will divide into two daughter cells, and the number of chromosome in each cell will be same. Means this is cell contains 46 chromosomes, so both the daughter cells will contains 46 chromosome. Means this somatic cell is diploid cell and it will form diploid cell. That's why it is called equational division. So this is called equational division. Why it is called equational division? Because the number of chromosome will be same in both the daughter cells. Now meiosis. What will happen in meiosis? It will take place in the gamete cells. There will be a number of chromosome in the cells becomes half. Like all the sexually reproducing organism, this meiosis will take place in the gametes. Means these are the gamete cells which meiosis will take place. Meiosis means what? In when the cell will divide, the number of chromosomes will be half. Why the number of chromosomes will be half? Because when the two haploid cells, or you can say these gametes, will fuse together, it will form a diploid. Means zygote is a diploid cell. Fuse together, it will form zygote. This egg is haploid and the sperm is also haploid cell. When they fuse, they form a diploid. See what happens in gamete cells, why the number of chromosomes becomes half. If the number of chromosome will be equal, like 46, if the number of chromosome will be 46 in the gamete cells also, if sperm contains 46 and egg also will have 46 chromosome, so what will happen? When the zygote form, there will be a 92 chromosome will be there in that zygote. So some other kind of organism will produce. So to maintain the number of chromosome in the organism, that's why meiosis takes place. In this, the number of chromosomes will be half. Means 23 chromosomes will be present in the gamete cell, so that it can form 46 chromosome, and the number of chromosome will be maintained in the organism. When two haploid cells will fuse together, it forms a diploid cell. Now we will see what is meiosis. This is a specialized kind of cell division that reduces the chromosome number by half, result in the production of haploid daughter cell. This kind of division is called meiosis. See this is a cell which contains 46 chromosome, that is a diploid cell, and it forms two haploid daughter cells in which the number of chromosome is half, 23 chromosomes. Meiosis is called the double division. Meiosis is called double division of a diploid cells. Means what? A diploid cell will divide into 4 haploid cells. In this, there will be a double division will takes place, And this meiosis is called reductional division. Why it is called reductional division? In meiosis 1, what will happen? A diploid cell will divide into haploid cell. Two haploid cells, the number of chromosomes become half. That's why it is called reductional division. This I told you, there will be two division will takes place in meiosis, and this is meiosis 1. In meiosis 1, the number of chromosome will be half, and it reduces. That's why it is called reductional division. And in meiosis 2, what will happen? These two haploid cell will divide into two daughter cells. The number of chromosome will be equal, means all the four cells will be formed, haploid cells will be formed, and the number of chromosome will be 23, 23, 23, 23. This is called equational division. Meiosis 2 will be called equational division. So meiosis will takes place in sexually reproducing organism. Formation of gametes. See, meiosis will takes place in sexually reproducing organism. There will be, this meiosis will takes place only in the formation of gametes. Third point is, meiosis consists of two division. Means what? There will be two division will takes place in meiosis. In meiosis 1, that is called reductional division, and meiosis 2, which is called the equational division. As I have discussed in mitosis, that before cell division there will be interphase. That interphase, what does it do? It is called a preparatory phase. This interphase is called the preparatory phase. It is same as place in mitosis before the cell division. In meiosis also, before the cell division there will be an interphase. What does means interphase? It means the cell prepare itself for the cell division. So what does it do? There will be G1 phase, S phase, and G2 phase. In G1 phase there will be a protein synthesis, RNA synthesis, so that it can synthesize, makes the proteins. And in S phase there will be a DNA duplication will takes place. Centrosome duplication will takes place. This centrosome, which will move towards the pole and forms the spindle fiber which get attached to the chromosomes, and it helps in the cell division. And in G2 phase also there will be a protein synthesis. M phase, when it enters, there is the number of chromosomes in the cell will be 46, but the amount of DNA will be double. The amount of DNA will be double. These two are called the homologous chromosome. See, these two will be sister chromatids, and these two will be non-sister chromatids. And when this homologous chromosome will divide in meiosis 1, just I am showing two chromosomes, so it will get divided into two cells, and the number of chromosome here, can you see the half number, means one chromosome is present in one cell, and the second chromosome will be present in the second cell. This will take place in meiosis 1. And what will happen in the meiosis 2? See this, I told you the content of DNA will be double in this. The content of the number of chromosome is the number of chromosome will be half, but the DNA content will be double. In this, what happened? Reductional division takes place, and again in meiosis 2 there will be a division, means this cell will divide into two daughter cells, and this cells will divide into two daughter cells. In this, the chromosome number will be equal in both the cells, and this is called equational division. Now I'll start with the M phase meiosis. What will happen? Just we have seen the interphase, and the cell will enter into the M phase, that is meiosis phase. So in meiosis, in the mitosis, what we have studied, there are the 4 phases. That is, first one, prophase, metaphase, anaphase and telophase. The phases will be same as mitosis. There will be 4 phases in meiosis. I am talking about meiosis 1. See, this prophase 1, I'll write it here, prophase 1, metaphase 1, anaphase 1, telophase 1. This prophase 1 meiotic division is longer. This prophase will be longer phase. This prophase is further subdivided into 5 phases. What are they? First one is liptotene. Second one is zygotene. Third one is pachytene, diplotene. Fifth one is dikinases. These are the subdivision of prophase 1. So we'll see what happens in this all the phases. In prophase 1 is more complex process because in this there will be a recombination of genetic material will takes place. In liptotene, this phase, the chromosomes we can see visible in this phase. There will be a compaction of chromosomes. Compaction means what? The chromosome will be start coiling in this and forms chromosome. It forms coiling. There will be a condensation of chromatin material into chromosome. During this liptotene phase, the condensation and coiling of chromatin fibers begin during the liptotene. And the second phase after the liptotene, there will be a zygotene. In zygotene, what happens? The chromosome starts aligning. The chromosomes will start aligning. In this zygotene, this stage, chromosomes starts pairing together, and this process of association is called synapsis. Means they will align together, they'll start pairing in the homologous chromosome. This chromosome synapsis is accompanied by the formation of complex structure called synaptonymal complex. The nymal complex in this which helps in aligning together the chromosomes. The complex formed by the pair of synapsed homologous chromosome is called bivalent or tetrad. This structure is called bivalent or tetrad structure. See, there will be, this is a synaptonymal complex. This is a chemical complex. This is a chemical complex. This is a chemical complex formed between the homologous chromosome. These two are homologous chromosome. And when these two homologous chromosome come together, they form a that is called synapsis, and this is called synaptonymal complex. So that these two, there will be a exchange of genetic material will takes place. There will be a recombination of genetic material will takes place. In zygotene, chromosomes are aligned. These two stages or phases will be for the shorter period of time, this liptotene and zygotene. After this phase, the third stage is pachytene. During this stage, the 4 chromatids of each bivalent chromosome becomes distinct and clearly appears as tetrad. Means 4 chromatids will be visible clearly in this. This stage is characterized by the appearance of recombination nodules. This is called recombination nodule. By the synaptonymal complex, these two chromatids will come together and attach here, and this is called recombination nodule. This is the site of crossing over means there will be an exchange of genetic material. Here there will be a exchange of genetic material will take place, and this exchange of genetic material will take place between the two non-sister chromatids. These two will be sister chromatids and these two will be non-sister chromatids. Non-sister chromatids of the homologous chromosomes. Crossing over is also an enzyme mediated process. This process will be takes place by an enzyme. That enzyme is called recombinase. These two non-sister chromatids will combine by the enzyme that is called recombinase. Crossing over leads to the combination of genetic material on the two chromosomes. Here like this, there will be an exchange of genetic material will takes place. So how it will look? The exchange chromatid and this the character genetic material will exchange. In this phase there will be recombination between two non-sister chromatids. These two are non-sister chromatids. This is a enzyme mediated process and that enzyme is called recombinase. The crossing over of the genetic material leads to the recombination of genetic material. If there will be no crossing over, then what will happen? All the organism will look same. There will be no difference between the individuals. Offspring will look similar to their parents. Next is in pachytene, crossing over will take place between two non-sister chromatids of the homologous chromosome. Crossing over leads to recombination of genetic material. Next is diplotin. After the crossing over takes place in the pachytene, in diplotin what happens? There will be a dissolution of the synaptonemal complex. Here the synaptonemal complex is present and in this phase this synaptonemal complex will dissolve. Dissolution of synaptonemal complex. And in this phase, after the dissolution of synaptonemal complex, there will be a separation will takes place between the two homologous chromosomes from each other except the site of crossing over. Except this site and this site, all it will start separating from except the site of crossovers. These egg-shaped structures are called chiasmata. See this, they will form X shape and this is called chiasmata. X shape structure will be attached at the point of recombination. This chiasmata, that is called chiasmata. Where the recombination takes place, it is joined there. That is called chiasmata. Synaptonemal complex dissolve. Formation X-shaped structure will form at the area of recombination called chiasmata. Next is dikinesis. See, I have taken only 2 chromosomes so that it will be easy to understand, because I cannot take all the 23 chromosomes, 23 pair of chromosomes. So how it will look after the recombination? The chromosome will look like this, because after the recombination. This is the final stage of prophase 1, dikinesis. This is marked by the termination of chiasmata. Chiasmata is joined at the point of recombination, so it will get separated from that phase. There will be a termination of chiasmata, and there will be a disintegration of the nuclear membrane. Nuclear membrane will start disintegrating. This nuclear membrane will start disintegrating in this phase. During this phase, the chromosomes are fully condensed and the mitotic spindle is assembled to prepare the homologous chromosome for separation. By the end of dikinesis, the nucleolus disappears and the nuclear envelope also breaks down. Dikinesis represents transition of metaphase. Last phase of prophase 1, there will be this is marked by the termination of chiasmata. Termination of chiasmata, disintegration of nuclear membrane, disappearance of cell organelles. Centrosome starts moving towards the pole. So after the end of the prophase 1, the next phase is metaphase. Next phase is metaphase. Metaphase 1. In this metaphase 1, nuclear envelope disintegrates and the chromosomes moves into the cytoplasm. Bivalent chromosomes align themselves on the equatorial plate. In this chromosome will align into the equatorial plate. This is spindle fiber. This is metaphasic plate. Chromosome align on the metaphasic plate. This is spindle fiber, centrosome. The bivalent chromosome align on the equatorial plate. The microtubules forms from the opposite poles of the spindle attached to the kinetochore. This is kinetochore, and the spindle fiber will attach with the kinetochore. Nuclear envelope disintegrate, chromosome moves into the cytoplasm. This is cytoplasm. Bivalent chromosomes align themselves on the equatorial plate. This is equatorial plate or metaphasic plate. Next is anaphase. Anaphase 1. What happens in the anaphase? Two homologous chromosomes, these two are the homologous chromosome, separates from each other. So it starts separating from each other and start moving towards the pole. Chromosomes containing two chromatids held together by the centromere. This is centromere. Two homologous chromosome separates from each other. They start moving towards the pole. Chromosome separates and move towards opposite pole to spindle fiber attached to it. Number of chromosome becomes half. Reduction in the number of chromosome occurs during anaphase 1. Now next is telophase. In this telophase 1, the chromosome reaches towards the pole. Spindle fibers disappear and nuclear membrane starts appearing. Produces two daughter cells, each containing a single nucleus. Nucleus of the daughter cells receives only one chromosomes from each homologous chromosomes. See, there will be a reappearance of nuclear membrane. Cell organelles will again come in that, and in this there will be the decondensation of chromosome will not takes place that much, because again it has to go for further cell division, that is meiosis 2. Chromosome reaches the pole, spindle fiber disappears. Nuclear membrane and nucleolus appear, produces two daughter cells, and both the daughter cells will have half the number of chromosome. Next is, after the telophase, the nuclear membrane and nucleolus reappear. Cytokinesis follows. The stage between two meiotic division is called interkinesis and is generally short-lived. Duration between two meiotic division is called interkinesis, and this interkinesis is followed by prophase 2. So we'll see what is interkinesis. So this is the stage between telophase 1 and prophase 2. It is the gap which exists between meiosis 1 and meiosis 2. Gap between meiosis 1 and meiosis 2. What happens in this? The chromosomes elongated but do not form chromatin fiber, and in this phase there will be no replication of DNA will takes place. The replication of DNA took place in the interphase. Now again there will be no replication of DNA will takes place. No replication DNA takes place. Protein synthesis will takes place in this as it is required for meiosis 2. RNA and protein synthesis takes place as it is required for meiosis 2. So we'll start with the meiosis 2. This meiosis is similar to mitosis. This meiosis will takes place. The first stage of this meiosis 2 will be prophase 2, and this prophase will immediately start after the cytokinesis. Cytokinesis means the division of the cytoplasm, usually before the chromosomes have fully elongated. Nuclear membrane will disappear by the end of the prophase 2. The chromosomes again become compact. First one is prophase. Chromatids, a univalent condenses. Second point, nucleus, nuclear membrane, nucleolus and nuclear membrane start disappearing. I'll make two cells in this. Nuclear membrane starts disappearing and the chromosomes condenses. This is the prophase 2. Now next is metaphase 2. Next is the chromosome align at the equator and microtubules form opposite pole of the spindle get attached to the kinetochore of sister chromatids. The chromosome will align on the equatorial plate. Spindle fiber will attach with the kinetochore. So what happens in the metaphase 2? The chromosome align themselves at the equatorial plate in the metaphase 2. Microtubules from the opposite poles extend towards the equator and attaches to the kinetochore of the chromatids. Next is anaphase 2. Anaphase 2. In anaphase 2, the centromere holding the two chromatids splits and allow the separation and movement of the two chromatids. So it will get separated, it will split and move towards the pole. Centromere holding, this is centromere holding the two sister chromatids splits. See, these two are the sister chromatids splits and allow the separation and movement towards the pole. Movement of two chromatids. Next is telophase 2. In telophase 2, the chromatids reach the pole and start uncoiling. The spindle fiber degenerates and the nuclear membrane and nucleolus reappear and 4 haploid nuclei are formed. This is the end phase in which two groups of chromosomes once again get enclosed by a nuclear envelope. This is nuclear envelope. Chromatids reach the pole and start uncoiling, becomes thin, nuclear membrane and nucleolus reappear and cell organelles. After the telophase 2, there will be a cytokinesis will takes place. And in the cytokinesis, there will be a formation of 4 haploid daughter cells. There will be a formation of 4 daughter cells. So after the formation of 4 different haploid daughter cells will be formed. Cells will be formed with the 4 different combination of chromosomes will be present in the different daughter haploid cells. So that's all for today. Thank you.